Okay, welcome back to PAE 341. Alright, still talking about trichotillomania, the treatments. That's where we stopped in the previous video. Being having a single life cycle, a direct life cycle rather, having a direct life cycle, a single treatment is adequate. A single treatment is adequate, meaning that we can use formalin, or what we call a formalin bath. We can use formalin bath. We get the fish having the parasites on the body. We get the fish, we dip it into maybe a bowl containing formalin in order to kill the parasites on the body. Okay, or we can use what we call a salt bath. Salt bath is having a salt water solution, okay, or a salt solution. Then we can dip the fish containing the parasites or having the parasites into the salt water, and then it will deal with the parasites immediately. Or we can use copper sulfate or potassium permanganate, and also flushing the system may help to reduce the infestation levels in the fishes. Alright, in your lecture note, I have the diagram of trichotillomania protozoan, showing the cilia, that is the organelle for movement, and the denticles. Okay, the magnification is about 400, so you can appreciate the organism better. Then the third disease, protozoan disease of the ciliate order is what we call chilodonellosis. Chilodonellosis, and is caused by parasites of Chilodonella species. This Chilodonella species is a highly pathogenic ciliate, an ectoparasite on the skin and gills of a wide range of fishes, tropical fishes, temperate fishes, that is, fishes that are found in the tropical areas, fishes that are also found in the temperate areas. So the parasite has a flattened ovoid shape and is up to 80 micrometers in length, and is covered by 2 rows of cilia which move it in a steady gliding manner over the epithelial cells of its fish host. So that is the movement. The cilia helps it in the movement, and it helps it to make a gliding movement over the cells of the fish. So heavy infestations of Chilodonella are often associated with poor water quality. There are some ponds you see the water to be smelling, and maybe water that has stayed over a long period of time. It could be an artificial pond or even a natural pond. Sometimes we reduce the water, introduce new water, introduce clean water. But when the water is not frequently changed or from time to time, it makes the water quality poor. It reduces the ability of these fishes to get even oxygen. So when there is poor water quality, there could be presence of many diseases as a result of, there could be presence of many parasites as a result of the low quality of water. Now, organisms like the carps, organisms like the salmonids and catfish are the species that are mostly affected. An example of this organism of this species is the Chilodonella hexastica. It's most likely to be problematic at low water temperatures and is reported to be a serious pathogen. In carps. There's another one we call Chilodonella piscicola. It infects cyprinids particular, and they can be found on some other fishes, but they can cause problems at higher temperatures. That is when the weather is hot, so to say. So fingerlings can be especially vulnerable. The younger ones, the younger fishes, possibly because of their nature, possibly because they're still trying to build up their immune systems, they're still growing, they're still building up their cells and nutrients and all that. So the mass development of parasites causes a much higher production of mucus and disturbances in the respiratory function of the skin. Alright, it makes the fish to be restless. It can rise also to the upper layers. It makes the fish to be restless and makes it to rise to the upper layers of the water. Its entire body is covered with a bluish-white coating and is particularly noticeable in the head region. So a smear in the skin surface reveals numerous individuals of the parasites. Damage to the skin may then be subject to secondary invasion by bacteria, fungi, and other pathogens. So it leaves the fishes to invasion of other pathogens like bacteria, fungi, and some other pathogens alike. Okay, these parasites also cause localized hyperplasia of the gill epithelium. The thin respiratory epithelium is covered by hyperplastic epithelium, and this drastically reduces the respiratory surface of the gills. What we mean by that is, the hyperplasia or hyperplastic epithelium means the abnormal growth of cells in the epithelium, abnormal growth of epithelium cells in the gills. Okay, and they could be precancerous or it could just be an abnormal growth without any cancer in it. So that is that for the Chilodonella species. We'll now move on to the protozoans that are flagellates. Remember I told you that the three groups are based on the movement of the parasites. The first one are the ciliates, the second one the flagellates, that is those organisms, those parasites that have flagella. And then we have the amoeba. So the flagellates, the flagellated protozoans, they are small parasites that can infect fish externally, that can infect fishes externally and internally. Okay, they are characterized by one or more flagella that can cause the parasite to move in a whip-like or jerky motion. The flagella are longer and more powerful than cilia and are always few in number. Okay, they are just like I said before, they are whip-like in nature, so that makes their movement, that speaks about their movement. So one of those diseases of flagellates protozoans are what we call histiopodosis. This disease is caused by heavy infections on skin and gills of fish by parasitic flagellates belonging to the genus Histiopodo. It's an important disease that can cause severe loss among farmed or ornamental fish worldwide for more than a century. And this parasite is regarded as one of the most damaging parasites among farmed salmon, and is probably a major cause of mortality among salmonid fries or salmonid fingerlings. So the flagellate that is perhaps best known as a serious fish pathogen is the Histiopodo necatrix. It causes the disease we call costiasis. So generally the Histiopodo species spread rapidly between hosts in fish farms, most likely by both direct contact or through free swimming parasites. It could be by direct contact of free swimming parasites. Then heavy infections may occur when conditions favor the parasites, like poor rearing conditions such as low water flow and high crowding densities, that is overpopulation, overcrowding, are considered particularly important. So when the fishes are overcrowded, there could be a chance that there could be infestation of these parasites in the water environment. So massive infections on skin and gills can cause epithelial hyperplasia or hypertrophy, and may result in severe or fatal osmoregulatory or respiratory problems. That is, it could make them it could make them to have the inability to regulate their salt and water balance, and then it could have respiratory problems. Clinical signs of Ichthyophilus species are an excess mucus production that forms a blue-gray or white film over the body surface and gills. They have also been described in several non-specific clinical signs of severe and prolonged species of the parasite infections, including flashing, lethargic behavior, listlessness, loss of appetite, and increased mortality. That could be restlessness, that could be, this fatigue and inability to engage in activities and all that. And then it could lead to, finally, mortality. So each of those species are obligate ectoparasites with a direct life cycle on the host, and they cannot subsist or multiply within an appropriate host. Without an appropriate host. That means their life cycle is centered on the fishes. So outside the fishes, they will not be able to complete their life cycles. So there are two forms. One, a parasitic feeding form, which attaches to the fish's epithelial cells, and a non-feeding swimming form of the parasite that exists off the fish. This one is non-feeding, so it exists outside the body of the fish, but the parasitic feeding form attaches to the fish epithelial cells where it gets its nutrients, of course. So the free living form is kidney shaped and it has two pairs of flagella. That means 4 flagella. The attached form is pear shaped and attaches to the gill and skin. The attached form is pear shaped and attaches to the gill and skin. The parasites disappear from a dead host and die after 30 to 60 minutes in the free swimming form outside a host. That is, the parasitic feeding form is the one that is pear shaped because it is the only one that attaches to the gills and skin. But the non-feeding swimming form exists outside the fish. So the one that attaches is the parasitic form, and is able to disappear from a dead host and dies maybe after 30 to 60 minutes in a free living form outside the host. So without the fish, it cannot exist and it cannot continue its life cycle. Then the next flagellate disease is hexametasis. Hexametasis is caused by excessive numbers of flagellated protozoa of the genus Hexameta in the alimentary tract of farmed and wild freshwater fishes. So the two species of the flagellated parasite Hexameta are Hexameta intestinalis and Hexameta salmonis. Both of them affect the gallbladder and upper intestine, including the pyloric cilia in young trouts and salmon in fresh water, and other aquarium fish in the warm period of the year. So the Hexameta species is a small pear-shaped, about 10 micrometers, piriform organism with 3 anterior and 1 posterior pair of flagella. That is 3 on the head side and 1 on the end side of the organism. Transmission is by ingestion of an infective cyst. In farmed Chinook and Atlantic salmon, the disease can become systematic, with fish becoming anemic with swollen kidneys. Being anemic shows that there is a possibility of being fed on blood and all that. So Hexameta salmonis may be present in vast numbers in the pyloric intestine of cultured salmonids in fresh water. So these parasites present a monogenic life cycle. The pear-shaped trophozoites change to spherical before cellular division. The pear-shaped trophozoites change to spherical shape before cellular division. They can be horizontally transmitted by the releasing of trophozoites and oocytes into the water from the fish feces that will be ingested by other hosts. So affected fish showed anemia, edema, inflammation, and congestion of the liver. Methods of prevention and control in this H. salmonis is by control of H. salmonis is by adequate water changes with the removal of organic debris. Constantly change of water. You don't need to leave your water to be so dirty and smelling. So when it's proper to change it, make sure you change it. And then and also remove the organic debris within the water that makes it most of the times green in color. So this is important in preventing infestation by the parasite. Now, an improvement in nutrition combined with vitamin C supplements can also be beneficial. You can use supplements to spray it in the water so that the fishes can consume them. Supplements with vitamin C and some other drugs that can boost the nutrition of the fishes. So number 3 is the velvet disease. This velvet disease is also called the gold dust or rust or coral disease. Okay, so it's a fish disease that is caused by a dinoflagellate parasite of the general Amylodinium in marine fishes. Therefore, it's found in marine water anyway, and Odinium in freshwater fishes, also in freshwater. So the genera that cause this velvet disease in marine water are called Amylodinium, whereas the genera that cause this velvet disease in freshwater is called the Odinium. Alright, the disease gives infected organisms a dusty brownish gold color on their bodies, and then the disease occurs most commonly in tropical fishes, that is, in tropical areas, and to a lesser extent, marine aquaria. Periodic use of preventive treatments like aquarium salts can further deter parasites. So regular monitoring, attentive care, and preventive measures collectively contribute to keeping goldfish health and velvet free. Okay. In your lecture notes that I sent across, you will see the diagram of the odium. I wasn't able to see the diagram of the amylodium, but you can check that out yourself. So this odium is a genus of parasitic dinoflagellates, just like I've said before. It causes velvet disease in fishes, and the life cycle is that the single-celled parasite life cycle can be divided into three major phases. One, the first is as a torment, and then as a termite, and then as a torment again. So first, as a torment, the parasite rests at the water's floor and divides into as many as 256 termites. Then the second is as these juveniles, the motile termites, swim about in search of a fish host. Meanwhile, using photosynthesis to grow and to fuel their search. Finally, the adolescent termites finds and enters the slime coat of a host fish, dissolving and consuming the host cells and needing only 3 days to reach full maturity before detaching to become a termite once more. So it goes from an adult termite, which divides itself into about 250 termites, and these termites grow to juveniles, and then as juveniles they grow into adolescent termites. Again, they bear the same name: juvenile termite, adolescent termite. Let me repeat: as a termite, as an adult termite, they go to the water floor and divide, just like we have binary fission, but this time around it's not binary fission because they're not two termites. So it divides itself into 256 termites, and then giving us the juvenile motile termites. So these juvenile termites swim in search of a fish host. Meanwhile, while using photosynthesis to grow and to fuel their search, so finally it turns into an adolescent. So the adolescent termite finds its way and enters the slime coat of a host fish. And when it does that, it dissolves and consumes the host cells, and thereby needing only 3 days to reach full maturity before detaching to become a termite once more. So what's the pathology? Vervets in an aquarium environment is usually spread by contaminated tanks, contaminated fish, contaminated tools such as nets and testing supplies. Okay, there are also rare reports of frozen live foods such as bloodworms containing dormant forms of the species. So frequently, however, the parasite is endemic to a fish and only causes a noticeable outbreak after the fish's immune system is compromised for some other reason. So the disease is highly contagious and can prove fatal to fishes. The symptoms: initially infected fish are known to flash or sporadically dart from one end of an aquarium to another, scratching against objects in order to relieve their discomfort. You see them when they swim across, maybe the edges of the pond, trying to, scratch against the walls or the edges, so that they can relieve themselves of the discomfort. Okay, they will also clamp their fins very close to their body and exhibit lethargy. So that's a feeling of maybe and maybe lack of interest in some activities in the water and all that. So if untreated, a dusting of particles, which are in fact the parasites, will be seen all over the infected fish, ranging in color from brown to gold to green. In the most advanced stages, fish will have difficulty respiring, that's difficulty in their respiration, and it will often refuse food. They won't be eating much, refuse food, and will eventually die of hypoxia due to necrosis of their gill tissues. So that means their death could be caused as a result of lack of oxygen, and it could lead to death of the tissues or cells of the gills. So treatment is by use of sodium chloride, which is our table salt or sea salt. This sodium chloride is believed to mitigate the reproduction of velvet in freshwater fishes. So however, this treatment is not self-sufficient for the complete eradication of an outbreak. Additionally, one of the common medications designated to combat the disease must be added directly to the fish's environment. These medications usually include the active ingredients of copper sulfate, methylene blue, formalin, and malachite green or acreflavine. Additionally, because velvet parasites derive a portion of their energy from photosynthesis, leaving a tank in total darkness for 7 days provides a helpful supplement to chemical curatives. So finally, some enthusiasts recommend raising the water temperature to an infected fish's environment in order to quicken the life cycle and subsequent death of velvet parasites. However, this tactic is not practical of all fish and may induce immunocompressing stress. May induce some stress because some species of fishes might not survive in water with raised temperature as that could kill the velvet disease-causing parasites. Okay. Now we move on to the third group, which is the amoeba. So in the amoeba group, several species have also been implicated. Number one is the amoebic gill disease. We call it AGD, amoebic gill disease. Amoebic gill disease is caused by the free-living facultative amoeba and is a major issue in marine salmon farming. It leads to gill damage and death of infected fish. It has become a significant problem in sea-caged Atlantic salmon and rainbow trout in Tasmania, and has been considered the most serious infectious disease. Cases of gill amoebic infections of fish other than salmonids have also been reported, for example, in European catfish or toboiled. Then clinical AGD most often occurs in water temperatures of 10 to 20 degrees centigrade and is sometimes associated with higher than normal temperatures. So gross pathology in infected fish is characterized by raised multifocal white mucoid patches on the gills, which present regions of epithelial hyperplasia of the primary and secondary lamella. So this phase is followed by discommission of the epithelium, local disturbances of blood circulation, and progressive changes represented by inflammation. So all the above mentioned changes result in decreased or loss of gill respiratory surface area. So the mucoid patches that can be found on the gills shows the presence of amoebic gill disease. So this disease affects just the gills of the host. It affects the gills of the host and therefore deals with the respiratory ability of that host, because gills is a respiratory organ for aquatic organisms and fishes inclusive. So significant cardiac changes and acid-base disturbances may occur in amoebic gill disease infested fishes, and it can lead to acute cardiac dysfunction and subsequent death of the organism or of the fish. So the second amoebic infection of fishes is the nodular gill disease. The nodular gill disease. We also refer to it as NGD. This is an infectious condition characterized by proliferative gill lesions leading to respiratory problems. They will always lead to respiratory problems because they affect the gills. Alright, so it can lead to respiratory problems. It also leads to oxygen deficiency and mortality in fishes. So anything that is able to affect the respiratory organ also affects the oxygen level or oxygen inflow into the body. So it leads to oxygen deficiency and then leads into death. That is the mortality. So globally, this NGD impacts freshwater salmonids and rainbow trout, the Oncorhynchus mycis. So in rainbow trout, with nodular gill disease, several species have been isolated from the gills, including several species of the nodular gill disease, including vanilla species, Negleria species, Protacantamueba species, Acanthamoeba species, and Hartmanella species. So how do we diagnose these parasites? Diagnosis is by macroscopic and histopathologic examination of the gills. So NGD is grossly characterized by multiple whitish nodular proliferations in the distal region of the gill filaments, often with diffuse white discoloration of the bronchial arches due to epithelial hyperplasia and or excess mucus production. So all these things surrounds the gills. The gill filaments, they are part of the gills. That is, the gill filaments are like the feathers or the most spread part of the gills. So all these infections affect the gills, and that's why it affects their oxygen intake. So clinically, fish show signs of respiratory distress and often high mortality. When there is high mortality, the death rate is too much. It means probably there's something affecting their oxygen intake, and it could be NGD. Alright, so concurrent infections with bacteria, mainly Flavobacterium species, are frequently detected on the gill surface of diseased fishes. So, what are the symptoms? Fishes can be less active, they can display respiratory distress, there could be changes in swimming behavior or reduced growth, and then mortality is usually noticed some weeks after the first symptoms appear. They usually die. And then the external aspect of the lesions in the gills is also characterized by the presence of whitish areas in the gill filaments and increased presence of mucus. Then, how do we treat this disease or infection? Most times the general treatments for ectoparasites remain the use of formalin, the formalin bath. It can be effective, but for NGDs, no specific data has been reviewed or has been made available about their treatment. But general treatment of ectoparasite of fishes, we use formalin, and we can also use osmotic shock. That is when you use salt solution. We dip the fishes containing these parasites in the salt solution so that they can receive osmotic shock that can kill or remove the parasite. So in fact, salt is one of the best, easiest, cheapest, and sometimes forgotten options used to treat certain ectoparasites and reduce osmotic stress in fresh water fishes. So that is that for the treatment, and I think that is that for the parasites. Of fish that we have for now. That is not all, but that is all we have for now as contained in the literature. So I want to thank you very much for listening, and I hope you do meet this piece well, and it serves you a purpose for your for impartition, and then I want to wish you well for your forthcoming examinations. I just want you to concentrate on maybe in the parasite of fishes. You should be able to describe a certain fish parasite, and I should be able to talk about the treatment, the life cycle. It's very important. Everything you know about it, according to the literature, just get one or two. So, and also you should be able to describe an ectoparasite of fishes like the leeches or the copepods. You'll be able to describe one of them, and then you should be able to at least talk about some of those enemies of fishes, at least one or two of them. You should be able to tell us how they can be controlled and all that. Just read the literature and just focus on these areas I just mentioned. Thank you very much.